Welcome back, guys. This video is for the second part of chapter 20, and I'm going to do something a little bit different here. I have an old video that I'm going to splice into this one, so you're going to see probably the format changes after a couple minutes from the beginning of the video, and it'll switch to an old video. So I may say something in the old video talking about seeing you guys in person, or an announcement for that class, but that's from last year or maybe even the year before that. But anyways, so I'll make this video here for the second part of chapter 20, and then I'll make a third video which is just for the identification of blood vessels. So let's get started. In this video we're talking about blood pressure, and blood pressure is the force of blood against a blood vessel. And usually what we're measuring, what you would measure like with a blood pressure cuff, is arterial blood pressure, which is the pressure in your arteries. And that makes sense, right? Because the pressure in your arteries is high, and then the pressure in your veins is low, all the way down to zero by the time you get to your vena cava. The arterial blood pressure pulses with the cardiac cycle. So it's highest when the heart is contracting and is lowest when the heart is relaxing. So that systolic pressure in your arteries is a pressure when your ventricles contracting. And then the diastolic pressure is a pressure when your ventricles are relaxing. And if you just take you feel your pulse anywhere, you can feel in your wrist or here in your neck, you can feel your pulse, and that's the pulsing caused by the contraction of your ventricles. That means that you're feeling your arteries. You can see a lot of your veins because the veins are typically more superficial, and that's partly because injury to your arteries is much worse than an injury to your veins because they are under pressure, so they would shoot blood out much more quickly than a vein would leak blood. Anyway, so when you're measuring blood pressure with a blood pressure cuff, what you're measuring is the pressure in the arteries. On average, an average person would have something like 120, right? That's the higher number, so that's systolic, and then over 80, which is the diastolic pressure. And so here's an example, of measurement through a blood pressure cuff. So you inflate the cuff, and this little cuff here gets full of air, and that air is pushing against the arteries here. And then you would slowly release the air by turning a little knob, and the air is going to slowly come out. And you would listen at the same time. And we would do this in person, but we can't do that. And a lot of you probably do this all the time for your jobs. And if you don't, you will in the future. But anyway, you listen here for when you can start to hear that pulsing. Because if this is the air pressure in the cuff is so high that you can't hear the pulsing, then the pressure in the cuff is much higher than the pressure in the artery. So you want to get to the point where you first can hear the pulse. That's going to be the greatest pressure in the arteries, so that's going to be the systolic pressure. And then you'd keep listening, and at the point where you can no longer hear the pulse, so right at the instant you can no longer hear the pulse, that pressure is going to be the diastolic pressure when the ventricles are relaxing. And another related term is pulse pressure, which is the pressure in your arteries that is added by the heart contraction. So if we take these same numbers, 120 over 80, if you subtract 80 from 120, that gives you 40, and it's measured in millimeters of mercury. So the amount of pressure that's being added into the blood by the contraction of the ventricles is 40. And that pulse pressure declines with age and also with disease. Another measurement we could see here is illustrated on this graph is a mean arterial pressure, or MAP. and the mean arterial pressure is the average blood pressure in your arteries. And you can estimate the mean arterial pressure by taking the diastolic pressure and adding a third of the pulse pressure. And you would do that because those are two things that are easy to measure. If you just take blood pressure, again, you get the systolic number and the diastolic number, and you can easily make this estimation with those two numbers. And if you look here, the top line is showing you the systolic pressure, which is of course higher, and then the bottom line is showing you the diastolic pressure, which is of course lower. And you can see that the pulse is occurring with the cardiac cycle going from high in systole to low in the diastole. And the mean arterial pressure is represented by this line that's going down the middle. Another few things you can see in this little graph is that the pressure is of course highest in the aorta because that's the artery closest to your ventricles. It would also be high in your pulmonary trunk. And then it's high also in your arteries, and as you get further and further away down to your arterioles, that pressure decreases. And then finally, by the time you get to your large veins, your pressure drops all the way down to zero in your vena cava, inferior and superior vena cava. The other thing you can see here is that right at this point, once you reach your capillaries, that pulse pressure is gone. So there is no difference anymore between the systolic pressure and diastolic pressure by the time you get to the capillaries. Okay, and I'm going to stop here in this video, and I'm going to splice this together with another video. It's going to pick up talking about venous return. I'll see you there. Remember that blood pressure is very high in the aorta and the other arteries that are coming out of the heart. And as you get away from them towards the elastic muscular arteries and arterioles, that blood pressure is dropping until you get all the way to the vena cava, where the pressure is effectively zero. So the venous blood pressure is so low that it's not driving blood through that system. So how does the blood get back to the heart? Well, that venous return relies on a skeletal muscle pump and a respiratory pump. And that skeletal muscle pump is something that happens in all of your skeletal muscles. Remember, your veins, larger veins, have valves in them that are similar to the valves of the heart. And these valves allow blood to go only in one direction, which is towards the heart. Whenever your skeletal muscles contract, they increase the pressure inside of your veins, and this causes blood to move inside of the vein. Because of the valves, blood can only move in one direction through the veins, from one little section of vein that's maintained by two valves to the next section. So the blood is pushed by the contraction of your skeletal muscles and ensures that it goes in the right direction by the valves that are in the veins. Similarly, the changes in pressure that are caused by breathing also move your blood back towards your heart. And this phenomena is known as the respiratory muscle pump. During inspiration, when you're breathing in, the diaphragm moves down and contracts. That increases the pressure here in the abdominopelvic area. That increase in pressure drives blood upwards into the thoracic veins. And even if it's driving some of the blood downwards, that blood doesn't really go anywhere because remember, these large veins have valves in them that cause the blood to be able to flow only in one direction, which is towards the heart. So anyways, when you're breathing in, that drives blood up here into the thoracic veins. When you breathe out, when your diaphragm relaxes, that causes an increase in pressure in the thoracic cavity, which drives the blood from the veins here in the thoracic cavity into the heart. It's also helping to draw blood up from your lower limbs into
the abdominopelvic area. So we've talked about the blood pressures that are relevant in the arteries, as well as how blood moves to the veins. So let's put some of this stuff together and figure out how you can know the total amount of blood that's flowing through the cardiovascular system. Well, the total amount of blood flowing is really dependent on two main variables. One is the pressure gradient, or the pressure that's given to the blood by the pumping of the heart. You can describe this using the MAP, the MAP, or mean arterial blood pressure. So in this case, this person has a blood pressure of a 120/80 and a mean arterial blood pressure of 93. In other words, this 93 millimeters of mercurial pressure is what's driving blood through the cardiovascular system. The flow of that blood through the cardiovascular system is opposed or resisted by a number of different factors. One is the viscosity of the blood. So the higher the viscosity, the more resistance. You can increase the viscosity of your blood through blood doping. And then there are a number of disease states which also increase the concentration of formed elements or blood albumins, which are going to increase the viscosity of your blood. The amount of resistance is also increased if you have a change in your blood vessel length. For the most part, our blood vessels don't change length significantly once we've reached adulthood. But if you gain a significant amount of weight, that can also increase the blood vessel length. That has a relationship to increase in blood pressure with an increase in weight gain. The main way that you control the resistance to total blood flow is by controlling the radius of your blood vessels. And if you remember vasoconstriction, or the narrowing of the lumen of particularly your muscular arteries and arterioles, that's going to lower the amount of blood that can flow through them, and it's going to increase resistance. The opposite will be vasodilation, where the lumen gets wider, and that's going to decrease resistance. And again, this is the main way that you control total blood flow, or the resistance rather, to total blood flow, by controlling the dilation and constriction of your muscular arteries and arterioles. And as we'll see in a minute, that's mostly controlled by the vasomotor center of the medulla oblongata, as well as by a number of hormones. So this pictograph is just showing you the same thing. The amount of blood that flows through the cardiovascular system is proportional to the pressure gradient, which is the pressure from the beating of the ventricles, divided by the resistance, which is determined by the diameter of the lumen, also by the viscosity of the blood, and the length of blood vessels. Of those, the factors that are going to cause an increase in total blood flow is if you have an increase in your cardiac output. That cardiac output, you'll remember, can be increased by increasing either heart rate and or increasing the force of muscle contraction in your ventricles. Vasodilation is also going to increase blood flow by causing there to be less resistance to the flow of blood. Okay, on the other hand, the things that are going to decrease blood flow include a decrease in cardiac output. This means you're lowering heart rate, or you're lowering the force of contraction, as well as vasoconstriction, which is going to provide greater resistance to blood flow. So there's a number of ways that the pressure of the blood is regulated. We can divide these into short-term and long-term regulation. Your brain provides short-term, very quick regulation. And this is the kind of change that happens in these instances where your blood pressure changes very quickly. For example, when you go from sitting to standing. The way that your blood pressure can be regulated involves changes in both cardiac output as well as changes in resistance. Major players in this system of the neuroregulation of blood pressure are baroreceptors. These are the receptors, as the name says. They're going to work by detecting stretch of the blood vessel wall. As you might imagine, if the blood vessel wall is stretched out more, that means blood pressure is up. If the vessel walls are less stretched out, that means the blood pressure is down. The major sites of these baroreceptors are in the aortic arch, which is, as you'll see, transmits information along cranial nerve 10, the vagus nerve rather, and it's going to have a main effect on your systemic blood pressure. You have another baroreceptor in the carotid sinus, which transmits on the cranial nerve 9 or glossopharyngeal nerve, and that mainly controls blood pressure to your brain, which is super important because you need that to be alive and conscious. These baroreceptors detect stretch of blood vessel wall, and then they transmit that information to the medulla oblongata, to a specific part of the medulla oblongata called the cardiovascular center. The cardiovascular center has two nuclei, the cardiac center, which can increase or decrease heart rate, as well as a force of contraction, and the vasomotor center, which influences vasoconstriction or dilation of blood vessels. So I kind of diagrammed that here, and then you'll see the picture from the book is on the next slide. But as I draw on the board, I just tried to simplify it here. So again, you have these receptors that are called the baroreceptors, one in the aortic arch, another in the carotid sinus. The aortic arch transmits information by cranial nerve 10, the vagus nerve, and that controls your systemic blood pressure. The carotid sinus transmits information along CN9, the glossopharyngeal, and it controls blood pressure to your brain. Both transmit signals to the medulla oblongata, specifically to the vasomotor and cardiac nuclei within the medulla oblongata. From there, the medulla oblongata has sympathetic nerve fibers, and I haven't drawn out, obviously, the preganglionic and postganglionic fibers. I just am drawing the overall pathway. Anyways, so the sympathetic fibers go to the SA node and AV node of the heart, and that, because these things determine the rate of your heart's beating, in this way you can change the heart rate. These sympathetic fibers also go to the myocardium, or the cardiac muscle cells, and that can change the force of contraction. Finally, sympathetic fibers also go to the smooth muscles lining your blood vessels to influence constriction or vasodilation. So you note on this diagram that the sympathetic output can affect a bunch of things: heart rate, force of contraction, and dilation or constriction. On the other hand, parasympathetic innervation is only influencing your heart rate by influencing the SA node or AV node. You can see that same thing on this picture, that the output of the sympathetic neurons, you can see it's color-coded here, goes to the SA node, the AV node, and then down here to the myocardium. Additionally, the sympathetic fiber goes to the smooth muscle of your blood vessels. On the other hand, your parasympathetic neurons, color-coded here, I believe that's red, are going to just the SA node and to the AV node. Here are the receptors that I mentioned, the baroreceptors that you have in your aortic arch, and a baroreceptor here as well. Oh, in the carotid sinus, sorry, I forgot to mention that. In addition, you also have some chemoreceptor reflexes, which mainly regulate respiration, but they do influence blood pressure as well. I included the same picture over here because these already picture these chemoreceptors as well, and it's very similar, the pathway to the baroreceptor reflexes. So you have chemoreceptors in the aortic arch, just like you had baroreceptors, and then you have carotid bodies, another type of chemoreceptor at the bifurcation of this common carotid artery here. As before, These receptors in the aortic arch send signals via the vagus nerve, and the carotid bodies send their signal via the glossopharyngeal nerve. What they're detecting is the level of carbon dioxide, pH, and oxygen. So what would lead to an increase in blood pressure? If you had high carbon dioxide, low pH, or low oxygen, this would cause the baroreceptors to stimulate an increase in blood pressure. It would also cause some changes in your respiratory system so that you would expire more CO2 and end up raising your blood pH. Okay, so both of those things, baroreceptors and chemoreceptors, are types of neural regulation that provide short and quick control over your blood pressure. You have hormones also that give you much longer term control, and they include angiotensin II, aldosterone, antidiuretic hormone, and atrial natriuretic peptide. We'll go into a little bit of detail, just a little, about renin-angiotensin system, and then the others I'll just give you an overall picture of how they work. Before I get into this diagram at all, let me just describe overall how angiotensin works. So if your blood pressure is low, or your sympathetic nervous system is stimulating your kidneys, angiotensin II is going to raise your blood pressure. And it does so by constricting your blood vessels, by increasing your thirst, and decreasing your urine output. And just to review that, you increase your thirst means you drink more. That adds more volume to your blood. Higher volume means a higher blood pressure. You decrease your urine, it means you're getting rid of less of that volume, and so you're going to retain more volume. And again, high volume is high pressure. Okay, so how does this work? Well, your liver all the time is producing this inactive hormone that's called angiotensinogen. That hormone is, as I said, continuously produced, and it's just circulating around all the time. If your kidneys detect low blood pressure or they're being stimulated by your sympathetic nervous system, they release another enzyme that's called renin. Renin then goes and converts angiotensinogen into angiotensin I, which is another inactive hormone. This angiotensin I is converted by angiotensin converting enzyme, or ACE, into angiotensin II, which is now that active hormone. And you may know there's a class of drugs that are called ACE inhibitors, and they inhibit this enzyme here. And so those ACE inhibitors can reduce your blood pressure or prevent your blood pressure from getting too high by preventing the action of this enzyme. Anyways, once angiotensin II is active, it's going to increase your blood pressure, as I said, by constricting your blood vessels, particularly the muscular arteries and the arterioles, by stimulating your thirst center, causing you to drink more fluids, and by decreasing your fluid loss, by decreasing urine output. So that's renin-angiotensin II. Overall, angiotensin II raises blood pressure. You have three other hormones we'll talk about. Aldosterone and antidiuretic hormone both maintain or raise blood pressure. And then ANP is the only one on this list that actively decreases blood pressure. Aldosterone is a hormone that's produced by your adrenal cortex. You may remember from when we talked about the endocrine system. And it works mainly by decreasing your urine output. It's not so different for the antidiuretic hormone, or ADH, which you may also remember is produced and released from the posterior pituitary, and is also maintaining or elevating blood pressure. It's increasing your water reabsorption in the kidneys, which means you're going to pee out less of that fluid. Stimulates the thirst center, and in large doses, it can cause vasoconstriction. Okay, and then the one that's different on this list, ANP, or atrial natriuretic peptide, is produced by the atria themselves, and it functions to decrease blood pressure by increasing urine output and increasing vasodilation. The stimulus for this is an increased stretch in the walls of the atria. So a protective measure to keep your blood pressure from getting too high. Okay, that's it. As far as the actual blood vessels themselves and naming them, I'll give you guys a list in class and in lab rather, and you'll start going over it there. And I will see you guys on Tuesday or Wednesday.